In episodes 1 and 2 of Peptide School, we talked about peptides as signals. We talked about what they are, how they're made, how they behave in the body. We applied that thinking to healing, to performance, to recovery. But today, the stakes change because when peptides are marketed, not just for healing, but for anti-aging, for regeneration, for longevity, we are no longer trying to fix an injury. We are trying to modify time. And biology does not negotiate with time without demanding something in return. This is Peptides 101, Part 3. And remember, knowledge is power. Hello, everyone, and welcome to the Reprogram Podcast. I'm your host, Dr. George Murphy. If episode 1 was about understanding the molecules, and episode 2 was about applying that understanding to healing and performance, then today is about something deeper. Because once you understand that peptides are signals, that they are instructions, not supplements, you're forced to confront a bigger question. What happens when we use those signals not just to repair damage, but to attempt to slow aging itself? That's a very different ambition. Healing aims to restore baseline. Longevity aims to alter trajectory. And that shift from restoration to manipulation is where discipline matters most. So before we go further, let's re-anchor the framework that carried this entire series. Remember, peptides are signals. Signals enter complex systems. And when you introduce a signal, 3 negative outcomes are always possible. Outcome number 1, nothing happens. Outcome number 2, something happens, but not what you intended. And outcome number 3, quality, purity, or sterility failures introduce real risk. Today, we're adding a 4th dimension, time, because longevity interventions aren't measured in weeks. They're measured in years, sometimes decades. And that dramatically changes how we should think about uncertainty. Okay, so why do longevity peptides feel so compelling? Healing peptides operate in urgency. Longevity peptides operate in fear. Fear of decline, fear of weakness, fear of irrelevance, and of course, fear of mortality. And when fear meets sophisticated biology, expectations escalate quickly. Longevity peptides are often described as cellular rejuvenators, telomere activators, mitochondrial enhancers, age reversal compounds. This is powerful language. It suggests precision. It suggests control. It suggests that aging is simply a dial waited to be adjusted. But aging is not a single dial. It's a network of trade-offs that evolved over millions of years. And evolution optimized for reproductive success is not lifespan maximization. That distinction is not philosophical, it's biological. Okay, so at the core of this entire conversation is a biological paradox. The same pathways that help you grow and repair in youth often accelerate disease later on in life. Now, isn't that interesting? Let's talk about the growth signaling axis, which we touched on a bit in episode 2 of Peptide School. So, growth hormone, IGF-1, a new one for you, something called mTOR, and cellular proliferation pathways. All of these systems are essential early on in life. They build tissue, they support reproduction, they repair injury, but they are not designed to run at maximum intensity forever. There's a concept in evolutionary biology called antagonistic pleiotropy. Wow, that's a mouthful. It simply means this: a pathway that benefits you early in life can harm you later. Higher growth signaling can mean increased protein synthesis, maybe faster repair, greater muscle maintenance, maybe higher cell survival. But it can also mean increased cancer risk, reduced autophagy or mitochondrial function, accumulation of damaged cells, or perhaps even metabolic dysregulation. There is no biological switch labeled regeneration only. When you turn up growth, you also influence this entire system. And the system includes mutated cells, precancerous cells, cells that were never meant to receive prolonged survival signals. Remember, cells do not know your intent, they simply respond to signals. Okay, now I want to introduce you to a new pathway we haven't directly discussed in this series as of yet. We've talked about growth hormone, we've talked about IGF-1, but there's another central regulator in aging biology that you'll hear about continuously if you spend any time in longevity research. It's called mTOR. And if that term's new to you, that's completely okay, because even within biology, mTOR only rose to prominence in aging science over the last few decades. So mTOR stands for Mechanistic Target of Rapamycin, another mouthful. That name comes from the drug called rapamycin, which was originally discovered in soil bacteria on Easter Island, also known as Rapa Nui. So rapamycin has actually led many lives. It was first studied as an antifungal compound, then as an immunosuppressant used in organ transplantation. But as researchers studied how rapamycin worked, they discovered something far more significant. It actually inhibited a fundamental cellular pathway, one that regulates growth, nutrient sensing, and energy availability. That pathway became known as mTOR. So, what does mTOR actually do? Let's simplify it. Think of mTOR as a master growth switch inside your cells. It constantly asks, do we have enough nutrients? Do we have enough energy? Are growth signals present? Is this a good time to build? When the answer is yes, mTOR activates growth programs. So that means increased protein synthesis, increased cell growth, increased cell division, reduced cellular recycling. When mTOR is active, cells prioritize expansion. So that's incredibly useful early on in life. It supports development, it supports muscle growth, it supports fertility, and it supports things like wound healing. You would not survive without mTOR. But here's where aging research changed the conversation. In yeast, worms, flies, and eventually mice, scientists repeatedly found something really unexpected. When mTOR signaling was reduced, lifespan increased, not slightly, significantly, across multiple species. That finding was profound because it suggested that aging might not just be passive wear and tear. It might also be driven, in part, by chronic growth signaling. So when mTOR activity decreases, cells shift priorities. Instead of building, they maintain. They increase something called autophagy, which simply means structured cellular recycling. So damaged proteins are broken down, old cellular components are cleared, stress resistance improves, maintenance increases, accumulated damage decreases, and over long periods of time, that shift appears to influence longevity. So now let's be clear. That does not mean growth is bad. It means growth has context. In youth, growth dominates. In aging biology, maintenance becomes much more important. So that's the paradox. The same pathway that helps you recover at 25 may, if chronically elevated at 65 or 75, reduce cellular maintenance and increase vulnerability to disease. Okay, now here's where rapamycin re-enters the picture, because rapamycin, the compound that inhibits mTOR, has also been shown in animal models to extend lifespan even when it's given later on in life. That finding has triggered cautious human studies exploring whether modulation of mTOR itself might influence aging biology. So notice this framing. Researchers are asking whether gently dialing down growth signaling might improve health span, not turning it up. That tension matters because many peptides marketed for regeneration, vitality, and anti-aging either directly or indirectly increase growth-related signaling. They raise IGF-1. They enhance nutrient sensing. They amplify anabolic pathways. And those pathways converge on mTOR, which brings us to an important question. If decades of aging research consistently show that reduced growth signaling is associated with increased lifespan in multiple organisms, what does it then mean to introduce peptides that may actually amplify those same growth signals later on in life. That doesn't mean those peptides are automatically harmful, of course, but it does mean we should understand what systems they are touching. And that brings us to the specific compounds often marketed as longevity enhancing peptides. Because once you understand mTOR, you start to see that many of the peptides are not operating in isolation. They are interacting with the very pathways aging research has spent decades trying to carefully understand and regulate. Okay, now that we've talked about mTOR, this master growth switch inside the cell, we can better understand what many so called longevity peptides are actually trying to do, because most of them fall into one of three categories. They try to stimulate growth, or improve cellular energy, or maybe even reset aging signals. So let's walk through a few that are commonly discussed, not to promote them, not to dismiss them, but to understand, as we've done before, what they're actually claiming to influence. Let's start with Epitalon. Epitalon is a short synthetic peptide modeled after a naturally occurring peptide from the pineal gland. It's often marketed as a telomere activator. So let's unpack that in plain language. So your telomeres are the protective caps at the end of your chromosomes. You can think of them like the plastic tips at the end of your shoelaces. So every time a cell divides, telomeres get slightly shorter. Over time, when they become too short, the cell can no longer divide safely. So that shortening process is one of the biological hallmarks of aging, which we've discussed before. Epitalon is proposed to activate an enzyme called telomerase, which helps maintain or lengthen these telomeres. So, in some animal studies, there were signs of telomere length changes and modest lifespan extension. So, that's really interesting. But here's that biological tension we talked about. Telomerase is tightly regulated in the human body for a reason. In most adult tissues, telomerase activity is very low. Why? Because unlimited cell division is one of the defining features of cancer cells. In fact, many cancer cells reactivate telomerase specifically so they can continue to divide indefinitely. Now, to be very clear, there is no solid evidence that Epitalon actually causes cancer in humans, but activating telomerase is not biologically neutral. It touches a system that the body normally keeps under very tight control, and long-term human outcome data simply does not exist. So, the question becomes, are we safely supporting cellular stability? Or are we modifying a boundary the body intentionally enforces? So that's not fear-based. It's just biological reality. Next, let's talk about MOTS-c. So MOTS-c is a peptide that originates from the mitochondria, those energy-producing structures inside all of your cells. So it's gained attention because it appears, at least in animal studies, to influence metabolism. So, in simple terms, MOTS-c seems to help cells manage fuel more efficiently. So, some studies in mice actually suggest it may improve things like insulin sensitivity and metabolic flexibility, meaning the body becomes better at switching between burning sugar and burning fat. So, that sounds really appealing. And metabolically speaking, maintaining flexibility as we age is really important. But again, most of the compelling data is from animal models. We do not have large, long-term human trials showing that MOTS-c extends lifespan, or reduces major disease risk, or meaningfully alters aging trajectories. So what we have is early-stage metabolic research, not proven longevity intervention. And here's the bigger point. Improving a short-term metabolic marker is not the same as extending lifespan. Biomarkers move faster than outcomes, remember. Longevity requires decades of observation. Another category often discussed is thymic peptides, compounds like Thymosin Alpha-1. So the theory here is really straightforward. As we age, the immune system changes. It becomes less precise, less responsive, sometimes even more inflammatory. So the idea is, can we rejuvenate the immune system with peptide signaling? So, in certain medical contexts, such as specific immune disorders, thymic peptides are studied and sometimes used. Now, again, that's very different from broadly administering them to healthy adults in the name of anti aging. The immune system is not a simple on and off switch, it's a balance between activation and regulation. Stimulate it too much, and you risk autoimmunity or chronic inflammation. Suppress it too much, and you reduce surveillance against things like infections or abnormal cells. Again, this is not a rejection, it's a reminder. Immune signaling is system wide, and long term modulation without long term data carries uncertainty. Now, let's return briefly to some of the biggest players in the longevity category: growth hormone signaling peptides. So, in longevity circles, these are often reframed as restoring youthful hormone levels. The language shifts from performance to actual age reversal. But raising growth hormone in IGF-1 does not selectively restore youth. It increases growth signals throughout the body. So we already discussed how these pathways converge on our new friend, mTOR, which means increased GH and IGF-1 may actually increase growth signaling in things like muscle, bone, connective tissue, and potentially in other cell populations. So, short term, this may actually improve your body composition, but long term, the biology becomes much more complex. Higher IGF-1 levels in large human populations are also associated with higher risks of certain cancers. So, this does not mean that these peptides cause cancer, but it does reinforce a consistent theme. Increasing growth signaling later on in life is not the same as increasing lifespan. Those are two different biological goals. Now, let's step back and look at the pattern that's emerging. Epitalon touches telomere biology. MOTS-c touches metabolism. Thymic peptides touch immune signaling. And growth hormone peptides touch systemic growth. Each of these systems evolved with tight regulation, and most longevity peptides aim to amplify or extend one of those systems. But aging is not caused by a single failing dial. It's the cumulative effect of many interacting processes, and modifying one system often influences others. That's why longevity biology is so complex and why certainty should make you cautious. Okay, so here is the honest scientific position. Some of these peptides are really biologically interesting. Some have promising early data, but we do not have long-term human trials showing that they extend lifespan or reduce cancer incidence, maybe prevent cardiovascular disease, or meaningfully delay things like neurodegeneration. These are the outcomes that define longevity, not improved labs at 3 months, not increased energy at 6 weeks. Longevity is measured in decades. And until we have that data, humility is not pessimism, it's discipline. So, if we step back from the individual compounds, from Epitalon, from MOTS-c, from thymic peptides, from growth hormone protocols, a pattern starts to emerge. The biology is often very plausible. The early data is often very interesting, but the timeline is short, and longevity is not a short game. That's the core problem. Because when we talk about healing, we can measure outcome in weeks. When we talk about performance, we can measure outcomes in months. But when we talk about longevity, we are talking about decades. Decades of cumulative signaling, decades of subtle biological shifts, decades of interacting systems adjusting to this new input. And that changes the level of evidence that's required. Improving a lab value in 3 months is not the same thing as reducing cancer incidence over 20 years. Increasing telomere length in a short study is not the same as extending lifespan safely. Improving insulin sensitivity in mice is definitely not the same thing as lowering cardiovascular mortality in humans. Longevity is defined by hard outcomes: cancer rates, heart disease, neurodegeneration, metabolic disease, all cause mortality. Those are slow variables. They take time to emerge. And here's the uncomfortable reality: we do not have multi-decade randomized human trials of these peptides in healthy adults. Not for Epitalon, not for MOTS-c, not for growth hormone stimulating stacks used for anti aging, not for broad immune modulating protocols in otherwise healthy people. That absence is not a minor technical detail. It's the central issue. Because when you interfere in growth pathways, when you influence telomere maintenance, when you alter immune signaling, you are not running a 6-week experiment. You are introducing a signal that may interact with aging tissues for years. And aging tissues are not neutral. They accumulate mutations, they accumulate damage, they change their responsiveness to signals, which means the same peptide may behave differently at 30 than it does at 65. That's not fear based, that's how biology works. And this is where responsible thinking becomes uncomfortable because without long term data, what we are really doing is participating in a live experiment, an uncontrolled one. That doesn't automatically mean it's wrong, but it does mean the burden of humility increases. And when growth signaling, telomere maintenance, and immune activation are part of the conversation, there is one variable we cannot ignore: cancer biology. And that's where we need to go next. So let's approach this carefully. Cancer is not a foreign invader, it is a breakdown in regulation. At its core, cancer involves two fundamental shifts. First, cells grow when they shouldn't, and next, they survive when they shouldn't. That's basically it: uncontrolled proliferation and reduced program cell death. Now, let's think back to the pathways we've just discussed. Growth hormone increases growth signaling. IGF-1 promotes cell survival and proliferation. mTOR increases protein synthesis and things like cell division. Telomerase extends the lifespan of dividing cells. So none of these systems are inherently dangerous. They're actually essential for life. They're critical during development, they're critical during wound healing, and they're critical during things like reproduction. But they are tightly regulated for a reason, because the body's constantly walking a line between regeneration and restraint. In youth, that balance favors growth. As we age, mutations accumulate in our cells. Most of these mutations are harmless, but some are not. And the body relies on multiple protective systems to do things like limit excessive growth, remove those damaged cells, and maintain surveillance. When we increase growth signaling later in life, we are modifying that balance. Now, let me be very clear. There is no conclusive evidence that Epitalon, MOTS-c, GH secretagogues, or thymic peptides cause cancer in healthy adults. But there's also no long-term data proving that they do not increase risk when used chronically in aging populations. And that is the honest position. And in aging biology, uncertainty matters because cancer risk is not determined by one signal alone. It's influenced by things like accumulated DNA damage, immune surveillance, metabolic environment, inflammatory tone, and growth signaling intensity. When you amplify growth pathways in tissues that already carry decades of accumulated cellular stress, the biological context changes. That same signal that supports regeneration in youth may now have different implications later on in life. That does not mean these peptides are reckless. It means that growth biology is powerful, and power without long-term data requires humility. Okay, so scientists worry about growth signaling in aging tissues not because they are pessimistic, but because cancer is fundamentally a disease of dysregulated growth. If you increase survival signals, if you increase proliferation signals, if you extend cellular replicative capacity, you are also influencing systems that cancer cells can also exploit. Again, influence is not the same as causation, but ignoring that overlap would be intellectually dishonest. And this is why longevity biology is so cautious about simply turning things up, because aging is not caused by too little stimulation alone. It is caused by accumulated imbalance, and balance is harder to maintain than stimulation. So let's step back and look at what this entire series on peptides has really been all about. So episode 1 was about understanding the molecules themselves, what peptides are, how they're built, how they may behave in your body. Episode 2 was about applying that understanding to things like healing and performance, separating mechanism from outcome, separating plausibility from proof, separating anecdote from real evidence. And today, in episode 3, we widen that lens even further because when peptides are marketed for longevity, we are no longer talking about recovery. We are talking about time. And time forces a different level of discipline. So, longevity is not about how you feel next month. It's about cancer incidence, metabolic disease, cardiovascular outcomes, neurodegeneration, all cause mortality. These are very slow variables. They require long periods of observation. They require humility. And most of all, they require patience. Peptides are powerful because, as we've discussed, they are signals. Signals do not act in isolation. They ripple through systems that evolved under constraints. Growth and maintenance exist in tension. Regeneration and restraint exist in tension too. Performance and longevity do not always point in the same direction. So that does not mean peptides are inherently harmful. It means they are not neutral. And when something is not neutral, clarity matters more than hype. So curiosity is really healthy. Innovation is absolutely essential, but certainty in aging biology is something we do not yet possess. If someone is selling certainty in a field defined by complexity and trade offs, they are selling something other than the data. This series was never about rejecting peptides. It's about thinking clearly before introducing new biological instructions into a system shaped by millions of years of evolutionary compromise. Because once you start modifying growth, once you start modifying cellular survival, once you start modifying time, you inherit the trade offs that evolution never eliminated. And that is why discipline, not excitement, is what protects long-term health. This is the reprogram. And remember, knowledge is power. I hope you enjoyed the discussion. If you did, please follow the podcast and join me on this journey of understanding and self-discovery as we navigate the fields of longevity, aging biology, regenerative medicine, and the emerging field of how and whether we can safely influence them.